Okay, I took the silly hat off. Let's finish this up real quick. Let's finish watching the rest of meiosis so we can look at meiosis 2. I used to call meiosis meiosis, or mitosis because it was so similar to mitosis and because it appeared to me initially that I had a meiosis going on and a mitosis. Well, what I didn't realize the first time I studied it was that the independent assortment and the crossing over occurred in meiosis 1, making it very different from mitosis, actually. So let's take a look at this, finish it out, and then we'll make a couple of comments about the lab and what you need to do.
After cytokinesis is complete, a second division, or meiosis 2, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis 1. In prophase 2, we now have 2 cells, each with 2 chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase 2, the chromosomes line up along the equator. This is different from metaphase 1, where the homologous chromosomes were lined up. In metaphase 2, each cell has only one of each homologous chromosome. In anaphase 2, the sister chromatids move away from each other along the spindle fiber. And in telophase 2, along with cytokinesis, we see the formation of 4 genetically different haploid cells.
Okay, so as opposed to mitosis, in meiosis we end up with 4 daughter cells, and they are not identical. They are not identical. They're all separately different. Everyone's different. Everyone's unique. In a female, in a woman, we don't have 4. We have 1 egg, 3 polar bodies. We do have 4 cells, but we only have 1 viable egg out of this and 3 polar bodies. In the human male, we produce 4 very different sperm. And not only that, we produce millions of them every day, and not a single 2 of them are the same. Every one of the millions we produce are different, each one. And that's what makes us unique, and that's why it's so important. So when you go to the lab and you do the work, looking at the different kinds of cells and tissues, one of the questions that you're going to be asked is going to be related to the Mendelian genetics and I'm talking about lab 6 now. And one of the questions is going to ask, when will Punnett squares not work? When will Punnett squares not work? The answer to that is on page 166 of your text. So there are several situations when Punnett squares will not work. And when we get into Unit 6, I'm going to be talking about blood work and codominance of the different antigens, of the different actually alleles. And so that'll be one of the times. But there are other times when Punnett squares won't work. And so you need to read page 166 of your book pretty carefully to see how that operates. Okay, so let's finish up. The lab for Unit 5 is your cancer and normal cell lab, where you will count the number of cells that are either in interphase, or like this one, or in mitosis, like some of these others, in whatever stage they're in. The fourth question that you are to answer has to do with describing the steps of the cell cycle. Describe them all, and describe each of the stages of mitosis. Don't worry about meiosis. But you need to know about meiosis is what Darwin never knew. And Mendel's law of independent assortment. You need to know that. Okay? Alrighty. So, do well on this and then get ready for Unit 6, because we'll be talking about some very strange things that happen with genetics. Did you know that we can make cats glow in the dark by inserting genes into them from sea anemones? Oh,